Good afternoon. I think that it is delayed probably. I have just 5 minutes from now. I have to hurry up. Before I start my presentations, I'd like to express my gratitude to the organizer of this workshop, giving me a chance to talk to here today. I'm a member of Marine Biotechnology Research Center in KIOST. The title of today is a study on bioactive metabolites from tropical marine organisms. My presentation was composed in 4 sessions: introductions and natural product research from Korean waters, and natural product research from tropical marine organisms, and conclusion. Probably first 2 sessions should be hurry up. I think this is not important to us. Probably a lot of things are explained by Professor Concepcion, right? Yeah. So just move to the next slide. And this is also explained just before. First important thing is natural product is very important in ocean science. A lot of products are useful for drug development and the other things. So we have to do marine natural product chemistry. Let's start the drug derived from marine organisms. During the preparations of this slide, I didn't add the pre-art. You know, the pre-art is very important product, but the pre-art was prepared the Filipino scientist, right? Oliveira, Professor Oliveira. He's now professor of University of Utah. Originally, he collected smell from Philippines, right? Yeah, so I didn't add this product because it should be presented from Filipino scientists. So I didn't add the pre-art. Anyway, a lot of drugs are derived from marine organisms, and also a lot of important toxins are also prepared from marine organisms. Some of them are important: tetrodotoxins, alkali acid, and saxitoxins, brevetoxin. Very, very important toxins and drugs are developed from marine. Several scientists in Germany, or some of the scientists in KIOST, tried to develop medicine from tetrodotoxins. It is initial stage of drug development. So let's move to next. I'd like to explain the marine natural product science in KIOST from Korean water. This is the important site for collections in Korean water. Koreans are located between the subtropical and the boreal waters. The Korean waters are also influenced by crucial current. When we choose the collection site in Korean water, usually we didn't go to West Sea. The West Sea was covered by soft and bottom sediment. We cannot see anything in this water. Usually we want to go to east side or south side. According to our survey, the result of natural product chemistry in Korea, around 50%, about 50% of natural products are isolated from Jeju Island. The resources are fluent in Jeju Island. During last 20 years, we produced a lot of results from natural product chemistry. These are isolated from our laboratory during last 20 years. Probably 200 novel products are isolated from KIOST lab. Let's move to the next. From now, I want to explain the tropical resources. Actually, the Tropical Overseas Research Center was established in 2000, but the beginning stage we cannot approach with easy. After 2006, we can approach and we can collect the tropical organism in that area. During the collections and during the study of natural product from tropical resources, we think about the big difference between the Korean organisms and the tropical organism. Probably there are big difference. During the isolations of a novel product from tropical organism, we compared the structural deviations and the concentrations and the diversity, something like that. The main purpose of our project is obtained the chemical diversity from biodiversity. Anyway, we can collect a lot of organisms from tropical area like this. This is pictures of collection from marine organisms in Micronesia. A lot of things are collected, and then some of them are moving to KIOST like this. And we can start the natural product research from tropical marine organism. I think this is the first result using tropical marine organism. Probably 2007, we can collect large amount of marine organism in the tropical area using our overseas research in Chung, Micronesia. This is the first result. And then the next slide shows isolations of manzanamine derivatives from sponges. Lots of natural products was isolated. And at the same time, we want to produce biodiversity by chemical synthetic method. Some of functional groups are changed by oxidations and the reduction method. So we can get the biodiversity from natural product. By this way, we pursued chemical diversity by using biodiversity. And also, this is another example. Bromotyrosines are also isolated by this compound, this sponges. And this is another example. Very, very interesting result. Yeah, the orchiniastatins are isolated from spongy Orchinia lamosa. Some of them are very, very active from tumor cell line. Can you see the last line of this? Adriamycin is a very, very famous cancer drug. This is sold in market. As a result, you can see the activity of adriamycins and orchiniastatins. According to this result, the isolated products are almost 1,000 times active than adriamycins. The next slide. This is from the same sponges. We can isolate different product. During the isolations of this product, there are some interesting stories in my laboratory. When we collected these sponges, we prepared the crude extract. One of the crude extract from this sponge is one of the most strong. The sponge was given to my students, probably 2010. I gave the sponge after 6 months. My students give me a result of this. However, we didn't get any bioactivity from this. Probably there is something wrong. We didn't realize at the time. Anyway, the students graduate. He get a job in pharmaceutical company. He left, and then we think about again. At that time, Dr. Yeonju joined our laboratory. She started our investigation of the sponges again, and then she got this result. It is actually minor compound. So we believe that this product is produced by this. This product is intermediate of this one. Now we are preparing a new research project. We want to know biosynthetic pathway from this to this. Probably the project will be launched and the project will be successfully. We can produce by the other process, and we can get another stages for our research. Please help us, and please help Yeonju Lee. Anyway, by this way, we got an interesting result from this, and then I want to show some interesting pictures. Fortunately, we can get a new route to access the Philippine waters. last month, we can we can approach the collection site in Philippines, collaborate with the University of Philippines Visay campus, Department of Biology, Professor will provide the campus, and we can access the General Megado. General Megado is a very important person to Korean people in Samar Island. During this 4 days, we can collect a lot of marine organisms. Most of organisms collected that area was reserved in Philippines. We cannot brought out right now. Anyway, during this way, we will try to do some things in Philippine organism. This is other pictures with Professor Campus and our scientists and the Philippine students collect a lot of things, and then to the next step. Hopefully, we try to we want to expand our collaboration with the Philippine BSS and you. And this is conclusion. A lot of things are prepared from sponges, and a lot of compounds have some special bioactivities. Because the time is not so much, so I want to finish my talk from now. Thank you.
Now we are open for discussion. Are there any questions for or comment? Oh yes.
Just a minor correction. I think the island there is Humonhon Island, Samar. Is it?
Samar.
Yeah, because it's the island where my mother was born.
Oh, really?
Humonhon Island, Samar.
Yeah.
Yes.
Yeah, we have a very interesting experience in that island. When I visit a small elementary school, a lot of children are interesting to us, and we can get some special memories in that island. Very, very kindly persons. Yeah, thank you.